That's a good idea, right? All right, good morning, everyone, and welcome this morning to the William Grif Bowen lectureship. I will start off by saying, unfortunately, my colleague, Linda Peterson, who was so excited to be here and to celebrate this lectureship, unfortunately fell ill, and so I'm afraid you're stuck with me. But first, to talk a little bit about Grif and his legacy in this community. So he actually got his start in North Carolina, and I think he got just about every accolade you could get as you go through high school, college, where he was Phi Beta Kappa salutatorian. Then he went to UNC for his medical school training, was AOA, and then finally kind of shifted a little bit to the west and landed here in St. Louis, where he did his training for internal medicine at WashU, and then served as chief resident at the John Cochran VA. And then completed his fellowship here under the era of Bert Sobel. So, and then in 1980, which to me doesn't seem that long ago, but my gosh, it's almost 44 years ago, huh? So, joined the Maryland Medical Group, where he was a very well-known and active practitioner of internal medicine and cardiology. Very well known in the community. He's also been very active in participating in many educational activities, both here through the university and at other sites. He's been the governor of the Missouri ACC chapter, the president of the Barnes Jewish Medical Staff. He's been the Neville Grant Award recipient, and in 2011 also received the Outstanding Teaching Award from St. Luke's Hospital. So he's really been an outstanding steward of education and of compassion and patient care. And certainly, we're all used to seeing him around here, so it's great to see him such an active part of the community here. I will also say that when talking about some of his personal interests, there was a word here that I had to look up to figure out what it actually meant. When he was an avid golfer, a numismatist. Now, if anybody else in here knows what that is, okay, all right, well, apparently I'm just not very cultured. So it's a coin collector, but I did have to look that one up. Odonophile, I know that one. So, but he's also had a passion for scholarship. He certainly has had, as said in here, he's got an amazingly devoted set of family, friends, colleagues, patients, and that's really why this lectureship is here, to sort of recognize all of his commitment and those who have recognized his commitment to the care of individuals, as well as to education and to continuing research. So really, it's wonderful to have this here. We're grateful to have this lectureship exist and to honor his commitment to those important tenets. So I also now get the pleasure of getting to introduce our speaker for today. And I think, we're really in for a treat as we have somebody traveling internationally to give our grand rounds today. And so this is Dr. Andrew Murray, who actually is originally from South Wales in the UK, and did most of his work initially studying biochemistry and metabolism at Oxford, and then went to the British Heart Foundation, where he really began getting interested in metabolism, studying mitochondria in diabetic heart disease, and diving much more into sort of metabolic regulation and cardiac phenotypes. And then switched even to ketone body metabolism at a time when not many people were studying ketone body metabolism. It's kind of become very hot now, but he was studying that in his PhD time, or his postdoctoral time. And then in 2007, he moved to the University of Cambridge, where he really embarked on a very innovative and kind of unexpected twist of how to study metabolism, and we're going to get a big flavor of that today, which is looking at lessons we can learn from changes related to high altitude, and how this can shift our understanding both of normal physiology, but also understanding pathophysiologic conditions from a totally different perspective. And I think he's shed a lot of amazing insights, and we were reminiscing this morning that when he first presented some of these concepts as a poster about 20 years ago, he said only about one person came to the poster. So, but things have really exploded. I think you're really going to enjoy what he can present today. There's some very unique metabolic and physiologic studies done at Everest Base Camp. So, without further ado, please join me in welcoming Andrew Murray.
Okay, thank you ever so much for coming today. It's a really wonderful to see you all here, and it's great to be in St. Louis, my first ever visit. So I'd like to begin with a few quick thank yous. Thank you to Dr. Schilling for the great introduction there. Thank you to Dr. Cates for the warm welcome last night. Big thank you to my very good friend, Dr. Peterson, for the invitation to be here. And thank you too to Dr. Bowen. It's a really great honor to give this lecture in your name, sir. Okay, so those are my disclosures. We're actually going to begin the lecture in one of the most extreme environments on Earth. You're standing on the summit of Mount Everest here, 8,848 meters above sea level. It's an extreme environment, but I hope to convince you today that it's not a remote environment, at least in my mind, but a fascinating natural laboratory in which we can ask questions of fundamental importance to our physiology here at sea level, but also of relevance to a great many diseases. So here, 8,848 meters, 29,000 feet. The atmospheric pressure is about a third of that here in St. Louis. The air is still 21% oxygen, but the lower pressure means that that challenges oxygen delivery across the lungs, into the blood, through the system, to the tissues, to the mitochondria. We'll hear more about them. And then you get this metabolic and redox challenge at the tissue level, and arguably that's the defining physiological feature at high altitude, tissue hypoxia. But hypoxia is not unique to high altitude. A great many diseases in their most severe form can result in some form of tissue hypoxia. We're looking at a critically ill patient here. He's also a climber, as it happens, but a critically ill patient in ITU. And hypoxia is a reasonably common feature here, but also, and I don't want to draw too many parallels or oversell the metaphor, you also see many features which he might share with climbers near the summit of Everest. But you also find low oxygen levels as a perfectly normal part of our physiology at the other end of our life's journey during development. So the fetus and the placenta developing in utero is experiencing low oxygen pressures. Now here we wouldn't term this hypoxia, this is normoxia, at least in most cases, it's normoxia for the fetus. But it really, hopefully, hammers home the point that within our biology we have the capacity not just to react to hypoxia, not just to respond to it, but also to carry out critical processes during our development under these low oxygen conditions. So we have it within us to deal with hypoxia. So we find low oxygen in all of these settings. So to return to high altitude then, and at least when I first developed an interest in this and when I was learning about altitude physiology as an undergrad student, it's very clear there's a very well-documented time-dependent response to low oxygen levels that a climber, such as our friend here, climbing towards the summit of Everest, might experience. And within minutes of ascending to altitude, you see a rise in heart rate, you see a rise in ventilation, especially upon exertion. Given enough time, and we're talking maybe a week or two, you might see a rise in hemoglobin concentration, red blood cell mass, increasing oxygen carriage capacity. And given a bit more time, in some tissues, such as skeletal muscle, you might find an increase in capillary density, although it's not absolutely clear whether this is angiogenesis and new blood vessel growth, or a wasting of muscle around the existing capillary bed. But nevertheless, all of these features have been well described, and all of them, to some extent, offset the challenge of oxygen delivery. But this classic picture, and at least when I started to approach it, says absolutely nothing about what's happening at the distal end, and in particular, nothing about changes in oxygen utilization. And although these features, to some extent, mitigate the challenge of oxygen delivery, they don't completely correct tissue hypoxia, even in climbers who are breathing supplementary oxygen. So there's still hypoxia in the tissue. So it stands to reason that there might be adaptations within the tissue that alter the way those tissues are using the oxygen. Now, thanks to a huge amount of work by a large number of people, but recognized in the 2019 Nobel Prize in Physiology or Medicine awarded to Peter Ratcliffe, Greg Semenza, and Bill Kalin, we now understand a great deal about how our cells respond to low oxygen. And this is a simplified view, but to a large part, this response is governed by the HIF pathway, the hypoxia-inducible factor pathway. And in most cells in our body, HIF1 alpha and HIF2 alpha are constitutively expressed, but they're also rapidly broken down in the presence of high levels of oxygen. And this is an oxygen and iron-dependent mechanism controlled by the PhD prolyl hydroxylase enzymes. So HIF1 alpha and HIF2 alpha are hydroxylated. They're tagged for destruction by the von Hippel-Lindau, the VHL protein, and have no transcriptional activity. So it's a little bit like if you've got a manual car, a stick shift car, revving the car engine with the clutch down. You're expressing HIF, but they're being degraded. When oxygen levels fall, when you hit this low PO2, the hydroxylation doesn't happen. HIF1 alpha and HIF2 alpha are stable now. They form dimers, heterodimers with HIF1 beta. And then, downstream of this, they control the expression of hundreds, if not thousands, of genes. So downstream of HIF1 alpha, we do in fact find a lot of metabolic genes. So most glycolytic enzymes, to some extent, will be increased in expression in response to HIF1 alpha. Conversely, there's work, and I'm going to show a little bit of this today, but not too much, but there's work showing that HIF1 alpha suppresses the gene PPARA, encoded PPAR alpha. This is a favorite gene of mine. It's a master regulator of fatty acid oxidation expressed in heart, liver, skeletal muscle. So when PPAR alpha is activated, you see enhanced fat oxidation. So what this is suggesting is that at least in the short-term response to hypoxia, but possibly more sustained under the control of HIF1 alpha, you see a switch away from fat oxidation, fat oxidation is a great pathway, but it's very oxygen hungry, towards glycolysis, glucose oxidation, lactate production. So a metabolic switch that decreases oxygen demand. HIF2 alpha, on the other hand, expression rises a little later, is thought to control a more sustained response to hypoxia. The most famous factor downstream of HIF2, and there's a lot of overlap between HIF1 and HIF2, but the most famous factor downstream is EPO, which encodes erythropoietin and drives enhanced red blood cell production. So collectively, this pathway alters both the systemic response to hypoxia and the local cellular response as well. So it's a critical pathway, and we'll come back to it a few times. Now, I live and work here in Cambridge, and Cambridge is a fantastic environment. It's a great place to raise a family. It's a great place to study. I work with fantastic students. We've got great facilities and amazing colleagues. But I'm from South Wales, right? It's one of the most mountainous parts of the UK, and you'll see what we're really missing here and what I miss about my homeland. The skyline here is flat, right? This is right at sea level. It's the flattest part of the UK. So why on earth would somebody working in Cambridge work on altitude? Well, believe it or not, actually, the University of Cambridge has got a long and storied tradition in high-altitude research, and one of the most famous exponents of this, this great hero of mine, is Sir Joseph Barcroft. And Barcroft was the professor of physiology in Cambridge around 100 years ago, in the 1920s and 30s. He's a really remarkable man, and he used, again, living and working in Cambridge, he used much the same kind of modalities to study hypoxia as we do today. So he constructed a hypoxia chamber in what is now our department. It's where our departmental library is. And exposed himself to low oxygen levels. He cannulated his arm. He lost a couple of arteries in the process. This was his research assistant, Sergeant Major Secker, who I think was really the sanity check on Barcroft's experiments, and would pull him out of these chambers if he was in danger of doing himself too much damage. But Barcroft also did field work. He kitted out this railway carriage in Peru and took it up through the mountains to Cerro de Pasco, up to the mining villages there. He was in a bit of an early debate with his Oxford rival, Haldane, about how oxygen crossed the lung. Haldane thought it was an active process and oxygen was pumped. Barcroft said it was purely diffusion. And so to win this debate, Barcroft took this carriage up and measured oxygen levels in his own blood and those of the people he met there. So he got interested in adaptation as well 100 years ago. Later in his career, Barcroft shifted his attention to the fetal circulation. And it's not clear whether it was Barcroft himself who came up with this, but certainly his work started to draw parallels between these scenarios. And it gave rise to the idea of Everest in utero. The idea is, at least Barcroft put it across, that the fetus developing in utero in pregnancy is exposed to the same low oxygen levels as the climber on the summit of Mount Everest. It's a remarkable prediction when you think that, we were still another more than 20, nearly 30 years before Everest was even summited. It was not clear that it could be summited. Certainly neither of these things have been measured. So, was he right? Well, work in the 1990s, carried out by my colleague Graham Burton in Cambridge and others, made the first measurements of PO2 in the human placenta. And within the first trimester of pregnancy, the first 7 to 10 weeks of gestation, Graham measured oxygen tension at around 20 millimeters of mercury. Just keep that figure in mind. But what is it like on the summit of Everest? So in 2007, thanks to a combination of serendipity and an extremely understanding postdoc advisor, I had the opportunity to join the Extreme Everest research group under the leadership of Professor Mike Grocott. Mike was an anesthesiologist at UCL in London at the time. He's currently in Southampton. And the idea that Mike and the rest of the team had would be that by conducting large-scale medical research at altitude, we could understand the factors that drive people to do well or less well. Who, in other words, who gets crippling altitude sickness and who is able to climb to the top of Everest, not effortlessly, but without using supplemental oxygen. There are a limited number of people who can do that and survive those low oxygen levels, thrive under those conditions. So with our first expedition, we took 200 members of the public, just normal, healthy individuals who signed up and wanted to come along to Everest Base Camp down here. And we carried out a whole battery of tests, exercise testing, cognitive function testing, probing different parts of oxygen delivery. And I was recruited as a very green postdoc to drive some of the metabolic studies forward on this. Then nested within that study, a smaller group, not including me, I should say, I was down here holding the fort bravely, but as a smaller group ascended higher up the mountain with the aim of reaching the summit of Everest, carrying out many of the same tests, including VO2 max measurements on the south collar of Everest. And the centerpiece really was the aim to take arterial blood gases in climbers on the summit of Everest. This had never been done. There had been measurements previously of alveolar PO2, but arterial PO2 was still unknown. So on the day the team reached the summit, the conditions were turning, and Mike, I mean, he's a great clinician, scientist, but he's also a fabulous expedition leader, and he dropped the team down to a feature called the balcony at 8,400 meters. So just below the summit, where it was a bit sheltered, they erected a tent, and he was able to get femoral artery blood samples from 4 of the team. So this was our tent here. So these samples were sealed up in tubes, put into a vacuum flask on ice, handed over to one of these chaps in yellow. These are our climbing Sherpas, and again, you'll hear more about the Sherpas later. And Pasang Tensing, our climbing Sherpa, ran 2 kilometers down the mountain in about 20 minutes to the blood gas machine to get those measurements. So what did they find? Well, here's the data, and I'll talk you through this shortly, but the headline finding really was the mean arterial pressure was just under 25 millimeters of mercury, so a quarter of what we have in our blood right now, and the range was around 19.1 to 29.5. The individual who was at 19.1, we believe that's still the lowest measurement ever made in a healthy adult human. And he wasn't just healthy, he was conscious, he was talking, he was taking femoral stabs from the other members of the party, so really quite remarkable, actually. And notably, very, very close to that measurement in the placenta of 20 millimeters of mercury, seemingly confirming Barcroft's hypothesis for 100 years back, right? So how are they doing it? How are you still conscious? Well, of course, you don't just helicopter to the summit of Everest. First of all, helicopters don't fly that high, but secondly, that wouldn't be a good idea. You acclimatize over time. And the way these mountains are climbed is almost like a siege, really. You ascend so high, you come back down, you acclimatize, you go a little higher, and so on. And so we can dig into the data a little bit more to get a bit of a clue about how this is happening. And so the white bars are showing you oxygen pressure here as you move from London to Everest Base Camp, higher on the mountain up to the balcony. And you can see the white bars are falling, so pressure is falling. As we know, that leads to hemoglobin to desaturate. This is the second bar here. So we can see saturation is really falling in hemoglobin. But at the same time, hemoglobin concentration is increasing thanks to the action of erythropoietin. So the third bar in here, hemoglobin concentration rises. And curiously, these things seem to cancel out. So you've got less saturated hemoglobin, but more of it, meaning that even up to about 7,000 meters of altitude, oxygen content, milliliters of oxygen per liter of blood, is the same as it is at sea level. But of course, pressure is low. As you go higher, of course, this starts to drop off, and climbers term this area the death zone. You don't want to spend too long there, right? It's not a great place for a holiday. But here in the death zone, you really do start to see oxygen content falling. But does that mean then the content is normal, therefore everything else is normal? Well, of course not, because pressure is low, and pressure is also important. It's the driving force to get oxygen into tissues. So for our climate then, they are still limited. It's not all a story about content. So in terms of the metabolic studies, our first measurements, we had the opportunity to use cardiac imaging and spectroscopy to understand cardiac function and energetics in our climbers returning from Everest. And this was work carried out at the John Radcliffe Hospital in Oxford at the MR facility there. And so as well as imaging, we were using phosphorus spectroscopy, looking at ATP and phosphocreatine levels as a measure of cardiac energy reserve. So remember, our climbers have been to altitude, and they've come back down essentially to sea level in Oxford now. They're completely normoxic. If anything, I mean, their PO2 is restored. If anything, they've got more oxygen because their oxygen content is going to be supernormal. What happens to their energy reserve? Does it go up? After all, this is the idea behind altitude training, isn't it? You go to altitude, you come back to sea level. You've got more red blood cells, more oxygen content. Well, perhaps to our surprise, we found, first of all, a lasting impairment in function. This is diastolic function. Systolic function doesn't reach significance, but there is a diastolic impairment in these climbers post-Everest. And incidentally, you don't need to summit Everest for this. If we run the same data for those who trekked to Everest Base Camp, we saw exactly the same finding. So diastolic function wasn't completely restored, and energy reserve was also suppressed. And what this really suggests is that as a process for our acclimatization, yes, we're increasing oxygen delivery, but there's a persistent alteration in our capacity to use that oxygen, so it persists even when we return back to sea level. Now, the good news is you recover. Say good news, I was one of the subjects. So the good news is that 6 months later, when we got everybody back, we saw restoration of the energetics and restoration of function. There were also some morphological changes as well, right ventricular hypertrophy, a little bit of left ventricular atrophy. And these are well documented in climbers. But this was the first measurements of altered cardiac energetics. So why were energetics impaired? Well, fortunately, we'd been sort of live to this idea, and we had a few investigations along the way. And this is my colleague Dan Martin using side-stream dark field imaging to look at blood flow through the sublingual microcirculation. It's a rich capillary bed there. And I'll play the movie, and hopefully you can see this. This is what you would expect in a subject at sea level, and you can see the blood flow through these capillaries now. So Dan made measurements on the same individuals pre-Everest and actually on the mountains. So this is the same individual now at sea level. I'll play it again so you can get your eye in. Sorry about the slight glitch in the movie there. And then this is the same individual at 6,400 meters, and hopefully you can see the blood flow through these central vessels here, how sluggish and thick and slow the blood is. Why would that be? Well, the increase in hematocrit is going to increase viscosity, so that could be part of the explanation. But also, equally, there could be alterations in endothelial function. Nitric oxide synthesis is oxygen-dependent, and NO bioavailability tends to fall. So we're seeing sluggish microcirculatory flow, despite the fact that the increased hematocrit should, in theory, be improving oxygen delivery. So what about the mitochondria? Well, this is our lab at Everest Base Camp, and poor Dan here, he's returning from the summit of Everest, and he's been poked and prodded, and now he's giving us a skeletal muscle biopsy from vastus lateralis at the top of the thigh there. And so we took biopsies, and we preserved them, and we flew them back to my lab in Cambridge, and we put them under the electron microscope. And hopefully what you can see here, this is again the same individual in London, and then after summiting Everest, and we can see an area of damage here. But if you look at these darker areas labeled M, these are the mitochondria. And what you can hopefully make out is that mitochondrial content is falling in the muscle here. Curiously, it's not a uniform loss either. You find these subpopulations of mitochondria in skeletal muscle and also cardiac muscles. So those that are deeper lying in between the myofibers, but also those along the sarcolemma. And it seems that we preferentially lose those along the sarcolemma. So it suggests it's a regulated process. So we're losing mitochondria. We lose about 20% of muscle mitochondrial mass. But we did a little more with the biopsies. We measured a number of metabolic pathways, and I had a particular interest in fat oxidation. And when we measured this enzyme, it's one enzyme in the beta-oxidation pathway for fat metabolism. It's a mitochondrial enzyme. But the activity fell by more than 50%, really striking suppression. So this outstripped the general loss of mitochondria. So mitochondria overall are falling, but in particular, fat oxidation is suppressed. And we speculated that this may be a beneficial adaptation to enhance oxygen or the efficiency of oxygen utilization in hypoxia. We had the opportunity a few years ago to follow this up in a unique case study. This is a patient in Naples, Italy, in the south of Italy, who had a unique loss of function mutation in the VHL protein. Now, if you remember back to my HIF slide, this is the protein that breaks down HIF. So this loss of function in VHL means that HIF is constitutively active. So he's living at sea level in Naples, but his HIF pathway is always active. So it's almost like his tissue is responding to hypoxia, even though there's no hypoxia there. So even from birth, he had a very high hematocrit and needs to undergo regular phlebotomy to keep his erythrocytosis in check. And this was the case right throughout his life. We carried out a number of exercise tests on him, and we saw, in comparison with the control, the index patient here had increased lactate, increased ventilation in exercise, but also a low end-tidal CO2 reflecting hyperventilation. And we had the chance to study his mitochondria, and here we saw, again, suppression of a lot of the pathways, including fat oxidation, but also altered mitochondrial morphology. So this was a really interesting case study to probe this pathway a little further. So that's all very well for skeletal muscle, but our story was about the heart and suppression of cardiac energetics. Well, we followed this up in rats, and we have hypoxia chambers for rats in my lab. We can take them up to Everest base camps, around 10% oxygen, for 2 days or 14 days. And we studied the mitochondria in my lab using this device. This is a very sensitive oxygen electrode. And we measure oxygen consumption under the control of different substrate-led pathways. And what we found was essentially nothing after 2 days of hypoxia, but after 2 weeks of hypoxia, suppression in pathways supporting fat oxidation, and those driven by complex 1 of the mitochondria. We'd seen a fall in complex 1 in our climbers' muscles as well. So it really suggests these were key pathways to follow up. Moreover, when we looked at PPAR alpha, that factor that controls fat oxidation, quite a number of its downstream targets were downregulated in hypoxia, suggesting this is the mechanism to drive the suppression of fat oxidation. Okay, but are these beneficial or detrimental changes at altitude? We haven't really answered that question. And to do so, we can really draw on a natural experiment that's taken place in at least 3 places around the world, where human populations have moved to high altitude and inhabited these regions for sufficient time to allow for the selection of physiological traits underpinned by genetic differences that support their capacity to live, work, and critically to reproduce at high altitude and pass those genes on. And this has happened in Tibet, on the Tibetan Plateau, in the Ethiopian highlands in East Africa, and on the Andean Altiplano. And these populations have all been studied to some extent. The Tibetan and Andean populations are the best studied. And it's really fascinating. This is an independent event in each of these locations. So you do find examples, clear examples of convergent evolution, similar genes under selection in these different regions. But also you find some stark differences. And the clearest difference between the Tibetans and the Andeans comes to arguably the most celebrated feature of our adaptive response, and that's hemoglobin. And in the Andeans, you find high hemoglobin concentrations shown here in yellow in men and women. You find high hemoglobin concentrations right throughout their lives. But in Tibet, and this is somewhat counterintuitive, you find a lower hemoglobin concentration at any given altitude in comparison with Andeans, or indeed lowlanders. So at any given altitude, the Tibetans, the adapted group, have got a lower hemoglobin concentration than you or I. That's slightly surprising. It means they've got a lower oxygen content in their blood. So you can see it's shifted, the histograms for men and for women is shifted over to the left. So they're carrying less oxygen. They have, and both groups, in fact, to some extent, have high nitric oxide levels. And in the Sherpas and Tibetans, this is associated with higher forearm blood flow. And so we postulate that the combination of a lower hematocrit, slightly lower hematocrit, it goes up at altitude, but it's just suppressed in comparison with us and with other high altitude groups, and the higher NO leads to better blood flow and maybe, as a result, better oxygen delivery. So what's driving all this mechanistically? Well, these 2 papers came out simultaneously in 2010 describing some of the earliest evidence for the genetic signals underpinning these features. And so using 2 different approaches, these groups both alighted on the same top target associated with low hemoglobin. And this was a gene called EPAS1. It encodes hypoxia-inducible factor 2 alpha, so that factor that increases EPO. So presumably this is a loss of expression, and in fact is associated with lower HIF2 alpha levels. So this came out both in Tibet and in both groups as their top signal. Now, where did this come from? Well, it's a really surprising origin. I put this in because this came up over dinner last night. There's a group, Rasmus Nielsen's group in California, were looking at the early origins of this EPAS1 and found that it was not closely related to the Han Chinese, a neighboring population, or indeed any other human population. And the postdoc working on this was getting a bit frustrated, apparently, and then it was suggested they might broaden their search a little further, and they found the closest relationship for this EPAS1 variant was not with human EPAS1 at all, but with the Denisovan variant. The Denisovans were a proto-human species known to be cohabiting with Neanderthals and early humans in Central Asia. And it's now been put across that there was introgression of the Denisovan gene variant into the Tibetan genome very early on. And supporting the idea that this might be beneficial at altitude there's archaeological evidence of Denisovan presence on the plateau, including a mandible that was found on the plateau dating back 100,000 years. So this seems to be actually quite an ancient variant that's undergone selection and has survived in the modern-day Tibetan genome. So really surprising origin. When I drilled down, I remember reading these papers with really great excitement, and when I drilled down into the second of these papers from Tatum Simonson and others, I was staggered because they also highlighted their second and third hits as being Eglin1. Eglin1 controls the expression of prolyl hydroxylase, encodes prolyl hydroxylase 2, and PPARA. So PPAR alpha, this fat oxidation gene. I thought I was the only one thinking about fat oxidation at altitude, and then Tatum put this out there and blew my mind. And so we find three key signals around the HIF pathway and the control of metabolism. So HIF2 alpha, EPAS1, PHD, and PPAR alpha. We've since been working with Tatum, and we've recently, her group, we had a very small part in this study, I should say, but has recently described a variant in EPAS1 in Andean populations that is associated with lower hemoglobin and protection against this excess erythrocytosis in this population. And when this was expressed in HEC293 cells, it led to decreased expression of HIF gene targets under hypoxia. So it's a loss of function mutation. It's a coding mutation. It's a different variant from the Tibetan variant. So a great example of convergent evolution on the same pathway. It's only very modestly expressed in Andeans. You'll see our numbers are lower for the gene variants here and only heterozygotes in the variant. Only modestly expressed, but essentially absent in all other human populations. In fact, this amino acid switch, we only see it in coelacanths, this primitive fish species. So it's a really rare variant, but it is under positive selection. And this perhaps highlights that this Andean group is still in a state of flux when it comes to the natural selection in response to altitude in comparison with the Tibetans. So what we find then is that some of these variants were under selection in all of these groups, Tibetans, Andeans, Ethiopians, EPAS1, Eglin1, there's variants. PPAR alpha, we don't, as far as we know, find any evidence of selection in the Andean group. We do in Tibetans and we do in Ethiopians, but this seems to be another key difference between the groups. So in 2013, we returned to Everest for Extreme Everest 2. And here the goal was not to climb the mountain. We didn't have an expedition to the summit this time, but to replicate our ascent to base camp. But this time to compare the response of lowlanders ascending to base camp with those of an adapted population. And we focused on the Sherpas. Now, the Sherpas are renowned for their prowess as elite climbers on the highest Himalayan peaks. This is Pasang Tensing, who was our lead climber in 2007, the same person who ran that blood gas sample down the mountain for us. Pasang Tensing is the youngest of 7 brothers, all of whom have summited Everest. His eldest brother has summited 20-something times. You know, so I mean, if that doesn't convince you of the power of genetics at altitude, I'm not sure what will. But they're really remarkable people. But in this study, we weren't interested in the elite climbing Sherpas. We were interested in them, but we took along with us normal, everyday Sherpas living in and around Kathmandu. They're of Sherpa heritage, 4 Sherpa grandparents, was all validated. But we took them up into the mountains, and none of them had been to altitude within 9 months of the study, and many of them had never been up to their heartland, actually. And so we looked at them, we carried out the same tests on both of these groups. So I'll show you this first, because I'm sure you're dying to see the, I never get enough of these movies, really, but you want to see the microcirculatory flow in a Sherpa at Everest Base Camp. So this is a little lower than the video that I showed you last time for the lowlander. That was at 6,400. This is 5,300 meters. And this is our lowlander. So you can see the blood flow at Everest Base Camp there. And then that's our Sherpa. Phenomenal, right? It's extraordinary blood flow. And you know, if you can, this is all published, so if you're interested, you can dig into the papers and see, you know, there's also differences in capillary density and diameter, vessel diameter, and so on. I mean, a clear adaptation there. But we were also interested in the mitochondria, and we decided to be a bit more ambitious this time, and we took our oxygraphs with us up to base camp. These are two former students in my lab, James and Alex. They had the great privilege not only of running the Everest Base Camp lab, but also being subjects. So they had to give their own muscle biopsies for the study, 3 each, you know. So somehow they sign up for this, you know. So they were really wonderful, you know. I mean, I'm just in awe of the work these guys did. So I'm going to show you a lot of data, and it will all be the same here. But this is lowlanders on the left, Sherpas on the right. And we studied the lowlanders in London prior to departure, then at Everest Base Camp when they arrived, and then we had the opportunity, because they were running the lab, to take a later biopsy 2 months down the line to look at longer-term responses. The Sherpas we studied in Kathmandu at their home altitude. It's a different baseline, but it is their home altitude. And then again when they got to Everest Base Camp. So they followed the same ascent from Kathmandu up to Base Camp and gave their samples. The Sherpas went home after this early time point, so we don't have the later signal. But you can see here saturation, oxygen sats, fall in both groups. So the Sherpas, like the lowlanders, are getting hypoxic. And when we looked at the muscle, VEGF is a HIF target. It's upregulated in hypoxia. Goes up in the lowlanders when they get to base camp, perhaps comes down a bit then. And it goes up in the Sherpas too. So their muscle is hypoxic. It is responding to hypoxia. It's not that they have no response. When we looked at fat oxidation and we looked at the expression of that gene, PPARA, we found that this was lower in the Sherpas. It doesn't really shift with altitude, the expression, but it's lower in the Sherpas in Kathmandu, and it stays low at base camp. This is a downstream target of PPAR-alpha. It's a fatty acid transporter into mitochondria, CPT1. That's lower in the Sherpas at baseline and stays low, and it comes down in the lowlanders. So what we suggest is that PPAR-alpha itself is not affected, but its transcriptional activity is suppressed. So what we're seeing is acclimatization in our lowlanders that essentially phenocopies the adaptation in the Sherpas. So we share some similarities in our response, which is curious. We looked a bit more at fat oxidation. We measured capacity, and this was lower in our Sherpas compared with the lowlanders. It does come down a little in the lowlanders, and when you suppress fat oxidation, or when fatty acid supply exceeds demand, you would expect then a backing up. And what we find in our lowlander muscle is this accumulation of long-chain acylcarnitines. These are lipotoxic fatty acid-derived metabolites that you find accumulating in the muscles of diabetic or obese patients in association with insulin resistance and poor mitochondrial function. And yet, the Sherpas who've got a lower constitutive fat oxidation seem resilient and seem not to experience this accumulation. And we've got some ideas of what might underlie this, but it's curious that they seem to have an alternative mechanism to dispose of that fat. When we measured the mitochondria, the Sherpas had a lower overall capacity, so we're looking at oxidative phosphorylation capacity here. But we also measure oxygen consumption in the absence of ADP, so no ATP synthesis. This is inefficient use of oxygen. We call it leak respiration. And this was lower in the Sherpas at baseline. It comes down in our lowlanders, but it's lower there. And it means that their mitochondria are more efficient, even just back at these lower altitudes. So their mitochondria, they're using more efficient substrates, and they're running more efficiently. What does all this mean when we look at energy metabolism? Well, when we measured phosphocreatine, and ATP levels show a similar thing, but the phosphocreatine energy reserve starts to fall in our lowlanders, and after 2 months is really suppressed. In our Sherpas, it goes up. We're taking them into a low oxygen environment, and yet their muscle seems to be supercharged. They've got enhanced phosphocreatine levels. I think perhaps suggesting that their metabolism is better tuned to this low oxygen environment. And when we looked at oxidative stress, there's also a redox challenge as well, of course, and we had a number of different markers. We find, and there's a big spread here, some of our lowlanders get a lot of experience, a great deal of oxidative stress, others less so there's no significant star here. And they certainly suppress this with acclimatization. But just look at the Sherpas. No Sherpa showed any sign of significant oxidative stress in their muscle. So they really are resilient to this environment. So a big goal of Extreme Everest, I mean, of course, was to study altitude, but also to relate this to patients. And after we got back, I teamed up with Dan again. So this was Dan who made the measurements of microcirculation. And we took a fellow on, Helen, anesthesiologist, to do a PhD with us to study some of the same mechanisms in critically ill patients. And Helen, I've got to say, did an absolutely remarkable job of, essentially the expedition where we had a huge team running all these measurements on the mountain, it was Helen. And she made measurements of mitochondrial respiration, microcirculatory flow in critically ill patients and asked how it was altered over time. So she recruited 21 patients in the intensive care unit, the Royal Free Hospital in London. And we had 9 elective hip surgery patients as our reference group. Such is the nature, of course, with ICU studies. We lost patients along the way. The mortality here was around 50 to 60%, which is not unusual for that unit, of course. But we didn't lose any of our reference patients, as you would expect. Helen sampled them within 48 hours of unplanned ICU admission, got biopsies, and then blood samples throughout the week, and then a second biopsy at 5 to 7 days, so we can look at the dynamic response to critical illness. So the headline figure is that in comparison with the reference group, there was no overall difference in maximum OXPHOS or electron transport chain capacity, ATP, phosphocreatine. Initially a little disappointing until we drilled down into the pathways. And what we found here was that although the overall capacity wasn't suppressed, fat oxidation was suppressed, and it was rapidly suppressed. Within 48 hours of admission, fat oxidation capacity was low. We were also able, and we didn't think we'd be sufficiently powered, but we were also able to detect differences between those who survived critical illness and the non-survivors. And to our surprise, we found the greatest suppression at 48 hours was in those who would ultimately go on to survive, suggesting to us that a better metabolic fitness to deal with critical illness and downregulate fat oxidation was protective. We saw a similar thing with complex 1 supported levels. I'm showing fat oxidation and complex 1 here. It's both the pathways we detected at altitude. And then between 48 and 7 days, we find the survivors do recover, but the non-survivors show this continual but slower decline. So same directional response, but a different dynamic. So this decline in fat oxidation was again associated with accumulation of lipotoxic intermediates, so long chain, medium chain, acyl carnitines, diacyl glycerols. So this is all hinting at a negative lipotoxic environment. And in our non-survivors, there was elevated plasma triacyl glycerol as well, showing this backing up of fatty acids. We also saw a number of markers of redox stress, oxidative stress, so higher levels of methionine sulfoxide, and lipid peroxides particularly were higher in our non-survivors than our survivors. So they're accumulating fatty acids, they're accumulating an oxidatively stressed environment, and then you get lipid peroxides accumulating as well. So we can see that this metabolic shift, beneficial in the short term, may be detrimental in the longer term as you accumulate these lipotoxic intermediates. So we think the fat oxidation and complex 1 capacity, they're suppressed at altitude and in critical illness, and they may represent hallmarks of recovery. And we have more metabolic data as well. We're looking now to scale this up and also take advantage of different oxygenation strategies in ICU patients to try to get to some of the mechanisms behind this. So there's ongoing work here. But I just want to briefly return to an idea I put across at the beginning. This is Barcroft's idea of Everest in utero, because the logical question then is, okay, well, if all of us in utero are standing on the summit of Everest, what happens to Everest in utero on Everest? What about the Sherpa women who are carrying babies very successfully through to term at altitude? How is that working? How low is the PO2 there? And pregnancy at altitude is a unique challenge, really. And birth weight, on average, falls by about 100 grams for every 1,000 meters you ascend. So you can see at altitude here, you really shift it. But there's relative protection in comparison with low altitude, where birth weight tends to fall. At high altitude, birth weight tends to fall, but there's relative protection in some of the high altitude populations. You also find an increased incidence of preeclampsia, gestational hypertension at high altitude, and again, relative protection with high altitude ancestry. And that goes for both of these main groups, Tibetans and Sherpas and Andeans. So we asked ourselves, how is birth weight protected in highlanders? Is it mitochondria? Is it antioxidant protection? Is there altered fetal metabolism? Is there enhanced maternal blood flow to the placenta? Is the placental development altered to improve oxygen diffusion? So I'm working in collaboration with a group here in Denver, Colorado, but also collaborators in Bolivia. And my colleague Colleen Julian and Lorna Moore in Denver have made measurements in 2 locations in Bolivia, at Santa Cruz, 400 meters, so low altitude, and again in La Paz at 3,600 to 4,000 meters there. This is a really amazing, amazing example of a large population living at very high altitude. And these are incredible measurements that Colleen made of uterine blood flow in these women. And you see the blood flow rises across pregnancy in European Andean groups, and we see this in the sea level group. But if you look at high altitude in La Paz, you can see that this rise in uterine blood flow is so much greater in the Andean population. It's almost double the blood flow to the uterus there. Remarkable signal of adaptation. And what we said now, I mean, if you look at just the convective oxygen delivery, the oxygen being delivered would seem to outstrip the demands of the fetus and the placenta. And the idea is that the way the placenta works is that the venous PO2 is critical here. It's a venous equilibrator, and propping up the venous PO2 supports diffusion to the placental mitochondria and across to the fetal circulation to support metabolism. So what happens to the mitochondria? Well, I teamed up with Lorna, Colleen, and Ramon there, and we've been collecting placental samples in Denver, at Summit County at Frisco in Colorado, which is 1,300 meters higher than Denver, and then again also in La Paz in collaboration with researchers there. And the placenta is quite unique in our hands in that we can actually cryopreserve the placenta with a medium that we've developed and retrieve intact mitochondria back at the end of the day. So we don't need to fly out with our oxygraphs, which is great because I have a wife and a small child at home now, so going off on expeditions is, I wouldn't be very popular at home. And to cut a long story short, I'll just show you some of the data. This is work really led by Katie O'Brien and Jenna Armstrong in my group. But when we get these deliveries, we get hundreds of placentas shipped in. The whole group really pitch in and help out here. So it's a great team effort. And so as you move from Denver to Summit County, we do find a suppression of mitochondrial oxygen consumption. This does fall with altitude, as we expected. But when we added in the group of adapted highlanders from La Paz, and look at the difference in altitude here, you're much higher in La Paz than either Denver or Summit, we find this remarkable preservation of placental mitochondrial capacity here. So we think that the better blood flow is supporting placental metabolism. So how is birth weight protected in, and I've underlined Andean highlanders, we haven't looked at this in Tibet as yet, but in the Andes we find evidence of enhanced maternal blood flow, altered placental mitochondria. Colleen's also got great data showing potential antioxidant protection there. And I'm absolutely certain these other features will also be protected. So I hope I've convinced you this morning that altitude, I mean, it's an extreme environment, sure, but it isn't remote. It is a great place to work, but not just for the adventure, but for the insights it can bring to healthy physiology, fetal development, but also potentially very sick patients as well. It's very short notice, but if you are interested in some of these ideas, we're hosting a meeting in Cambridge next week. I realise it's a stretch for you to get to Cambridge, but it's free. It's free and it's online. So if you're at a loose end on Monday and Tuesday next week, we've got a great meeting on pregnancy at high altitude that we're hosting there, that the Royal Society is funding. So it's going to be a discussion meeting, so lots of chance to throw these ideas out. But really, this is the most important slide. I mean, there's a huge amount of work there, and really, I'm so fortunate to have such a wonderful international team of collaborators, fantastic students and postdocs and fellows in my group over the years, clinical collaborators. It's just been a great journey along the way. A special thanks to Mike Grocott and the Extreme Everest team. And Mike really put his faith in me as a junior postdoc to lead a lot of this metabolic work. And it's, the best journey of my career. And of course, all of our funders and the societies who support us. So just to finish really then by thanking you for listening this morning. And I'll put up this last slide, which I believe you need. So thank you very much.
All right, so we have time for some questions. All right. You're gonna make me run up there, huh? That's fine. Go ahead and yell it out. You're loud, Ali. Go ahead.
Questions at altitude. It was a great talk, really, really interesting. Kind of two questions. Really intrigued by the diastolic dysfunction that occurs, and kind of two related questions. One, do you think that that's occurring with the return? Because it strikes me as amazing that people are able to exert themselves at such high levels climbing.
Yep.
Right, without any real issues, and then And
Yeah.
Secondarily, it's surprising to me because what's the actual PO2 in the heart, and how much does altitude actually affect the tissue level myocardial oxygen content?
Okay, yeah, those are really great questions. So the question was about the diastolic impairment. Is it occurring specifically upon return to sea level, or is it present at altitude given they exert themselves? The second question was about the cardiac PO2. I can't definitively answer either, but with the return to sea level and the diastolic, I think you hit on a really important point here. With the reoxygenation will itself be a redox stress. But of course, this is energy dependent. I think I can answer this we can look at some great data on VO2 max at altitude, and that falls. And it falls in everybody. At Everest Base Camp, you're down to about 35% of your VO2 max. Climbers always feel that they're much fitter and healthier when they acclimatize, but actually when you measure the VO2 max, even after a few months at altitude, it's still 35%. They're no fitter. You can reoxygenate as well. There were cool experiments done in the 70s where they were given gases to breathe to restore PO2. And VO2 max goes up, but it doesn't go all the way back to sea level values. And that was really hinting at this peripheral impairment. We do find the mitochondria is suppressed. And of course, diastolic function isn't passive. It's very energy demanding as well. So I think there is a link with the ATP there and the energy levels being lower, although, so we haven't measured diastolic function at altitude is the bottom line though. And in terms of the cardiac PO2, I don't know the PO2 exactly. When we've looked at the rat hearts, we do find HIF is stabilized, which would happen below a critical PO2. So the heart is hypoxic. There's been various numbers that have been suggested around even mitochondrial PO2. And mitochondrial PO2 would be, down to sort of single sort of figures there. And how much room is there to suppress that? And I think that's an interesting question as well. So, but at least at the gross cellular level, PO2 is lower in terms of the HIF response. So.
Great. Samantha?
That was just a wonderful presentation. In heart failure, we see a reduction in fatty acid oxidation.
Yep.
Parallel to what you showed, there's been a debate as to whether anesthetics are protective or not in heart failure.
Yep.
If you do an analogy with your data, it suggests that this is a cardioprotective response. Can you give your thoughts on that?
Yeah, no, it's really great. And that was very much, the question was about fatty acid oxidation in heart failure. And fatty acid oxidation is suppressed, as we know, in heart failure. There's been a long debate in the field about whether this is adaptive or maladaptive. And it's a trade-off, right? It's, we store most of our fuel in the form of fatty acids. Fatty acids are more calorie dense. If oxygen's not limited, fatty acids are a wonderful fuel, but then, we can learn lessons from exercise as well, that at lower intensities, we might burn more fatty acids. As we can switch to higher intensities, oxygen becomes possibly more limiting, and you switch over towards more glucose oxidation and so on. And so I think there are parallels there. And I think for the failing heart, it's going to be a bit of both, right? And it's the switch away from fat oxidation is going to conserve oxygen availability and potentially be protective. But longer term, you're at the risk of accumulating these fatty acid intermediates and driving insulin resistance. We do see that in later stage heart failure. That was, I did some PhD work on, my PhD work more than 20 years ago was on some of those clinical models, and we were seeing insulin resistance occurring there as well. So you're at the risk of, if you suppress fat oxidation, of driving this lipotoxic environment. So yeah, absolutely. And I mean, these are, yeah, trade-offs. And I think what's curious with the Sherpas, and we do see in the lowlanders that accumulation of the long-chain acyl carnitines, the protection in the Sherpas is really intriguing. And there are some interesting ideas there in some of the genetic studies of alternative pathways that may be upregulated to deal with that. So we're forever learning from this data. So.
Back there.
Yeah, thanks for this incredibly compelling lecture. You showed data that high altitude impairs some aspects of energetics, decreased diastolic function, and microcirculation problems, and decreased skeletal muscle mitochondria, and yet, essentially every elite endurance athlete trains for several months a year at altitude.
Yep.
With the premise of increasing oxygen carrying capacity, et cetera. So how do you square this?
Yeah, I mean, so they don't train on the summit of Everest or even a base camp, right? So they'll train at more modest altitudes. But actually, I mean, that data is not as convincing as the sort of popular image would lead you to believe. It's not absolutely clear that training at altitude improves performance. There can be detraining. And so this idea of going to the altitude training camp and staying there for a few months and training can lead to a bit of deconditioning. So people have switched strategies to sleep high, train low, so sleeping in a hypoxic tent instead, and then training at competition altitude to maintain intensity. Conversely, people have done the other switch where you sleep low and train high and then have a hypoxic stimulus during the exercise training to try to promote adaptations. And there's not, I mean, a lot of those a lot of those experiments are not terribly well controlled, actually. I mean, the control will be, if somebody goes off at a training camp and comes back fitter, that shouldn't be a surprise. They've been training, right? So what you really need is the sea-level training camp as the control. And when those studies have been done, and there was a study about 10 years ago from a Danish researcher who was doing the live high, train low, he actually found no difference in VO2 max, in exercise efficiency, even in hematocrit, with the sleep high, live high, train low protocols. So the jury's still out on some of those protocols, and it could be, a lot of it could just be people going off on a training camp in the mountains and focusing and so on. But the Sherpas, when you bring them down to sea level, and the evidence, the rationale for doing this, part of it comes from the fact that you see the Ethiopians and Kenyans winning marathons at sea level. They have other adaptations. They have biomechanical adaptations to exercise, really strikingly. If you bring Sherpas down to sea level, they're fairly unremarkable, actually. They're not winning marathons. I mean, I'm not being rude. They don't win marathons at sea level, but there is this incredible marathon every year, the Everest Base Camp Marathon. I mean, I don't know, Dr. Cates is a runner, I hear. I think he's left, to see, but maybe we should sign him up. But this Everest Base Camp Marathon, the top 20 finishers are always the Sherpas. They're incredible performers at altitude, but kind of unremarkable at sea level, which is surprising. So.
We talked last night a little bit about SGLT2 inhibitors.
Yeah.
The effect they have on diastolic function, oxidative stress, mitochondrial function in the heart. Could you comment briefly on that?
Yeah, absolutely. And I mean, the question was about SGLT2 inhibitors and the effect on diastolic function, metabolism in the heart. I mean, clearly this has just been a remarkable story. And I mean, I'm not a clinical cardiologist, but I have colleagues saying, it's just changing practice. I mean, so we've had a meeting here in St. Louis the last few days, the Society for Heart and Vascular Metabolism, and so it's a wonderful society, and Dr. Peterson was organizing this great meeting. And 20 years ago at this society, we were really banging the drum for metabolism, saying, treating metabolism is going to be key to, the next therapies for heart failure, diabetic cardiomyopathy, and so on. And the SGLT2 story, and also, I mean, the GLP-1 receptor agonist story is completely vindicated that approach. Treating metabolism to treat heart failure is critical. The big question, I think, is still how they're working, and is it purely a renal effect, or people have put across the idea that it drives ketosis. Ketone bodies are modestly elevated. The really there's some reasonably compelling data now that there may be a direct effect on the heart through sodium-hydrogen exchanges and lowering intracellular sodium and improving mitochondrial function that way. And I think it's a fantastic story to follow. I mean, not just from a clinical point of view, but from a basic science point of view, it's a great problem. So, yeah.
Fantastic. Well, I think with that, we'll let Grif have the last word on that with that great question. Thank you, everybody, for tuning in. Thank you, Dr. Murray, for a wonderful lecture, really insightful. And again, thanks, everybody, for attending.